Welcome to BioStock. Today we're putting a focus on mental health. Gabather is a biotech company based in Stockholm, developing treatments for mood disorders like schizophrenia or depression, among others. Here to present the company and its pipeline is CEO Michael-Robin Witt. Thank you for joining us, Michael. The floor is yours.
Thanks a lot, Michael. Great to be actually back at BioStock here in Lund, and I'll give you a presentation of Gabather, a little bit of a snapshot of where we are today, and also a little bit of how we work and what our philosophy and what our ideas are, how to move ahead with next generation in mental health. First part of the presentation is just a presentation of the company, and it was actually founded based on research from Lund University, and actually from Lund University, not very far from where I'm right now. So it's quite a journey this company has gone with this project and its ideas. Our area, as already mentioned by Michael, is in drug development in mental health. We focus on small molecular weight compounds. And very important, I think, to mention is that we do develop candidate drugs from bench to bedside. So we synthesize our own molecule. We do not license in and license out, but we produce our own candidates from the very beginning all the way to the bedside, i.e. in a clinical setting. I'm going to talk about much more today. We're going to talk a lot about our lead substance, lead candidate GT002, which right now is in the clinical trial stages, actually in the third clinical trial right now that is ongoing in a so-called target engagement study. I'm also mentioning a little bit about the patterns that we have, the pipelines that we're working on, and of course the reason for presenting Gabather, that we're currently going through a rights issue transaction, and we're looking at extending our runway for Gabather to continue our project, which I'm going to present now. Mental health, as mentioned, is an absolutely enormous and one of the largest health issues of our times. It is actually increasing, not only due to the increasing age, but also increasing societal events, effects. Things like social media, for example, have a big impact on mental health, particularly in young people. And as you can see on this slide, the societal impact is huge. The social economical impact is even larger. It's very difficult to guess this, but in all these figures, we're talking actually billions. The lack of innovative medicines is actually, that's why I have a question mark behind this bullet point, is actually quite critical. And if you go out and present to, for example, patient groups, and the first question that you always will get as a person that works in drug development is, when will you guys invent something new, come up with something new? We're trying all the medicines, none of them work as we want them to do. So the large unmet medical need, which is basically just there's a need for new medicines, both from the patient side and also from the therapy side, of course, from the doctors. They're looking for new therapeutic approaches. And that's what Gabather does. We work on a neurotransmitter system, a modulator system in the brain called GABA, stands for gamma-aminobutyric acid. The summary of GABA and the key take-home message is that GABA is the key inhibitory transmitter system in the brain. The brain is regulated by excitation and inhibition, by acceleration and depression, and glutamate is the accelerator, as you can see in this slide, and GABA is the inhibitor, the brake in the car, if you wish. So what we do with our drugs and our drug candidates, we enhance the inhibitory activity of nerve cells through the GABAa receptor modulation, and we have good preclinical evidence that we actually are able to relieve cognitive symptoms and cognitive deficits, at least in preclinical models. And that, I think, makes it a very interesting drug candidate and a very interesting drug development project, and a very much needed one. And the reason for that is, as you can see in the next slide here, is that I have put on the top row a number of neuropsychiatric diseases, and on the columns I put what symptoms are associated with this kind of disorders. And you can see they're, of course, all a little bit different. What goes through all of them, and is in common to all of them, is that all have some form of deficit in cognition. And of course, the most extreme and the most obvious one is things like Alzheimer's dementia, where cognition, i.e. deficits in memory, deficits in learning, are so enormously reduced that become really incapacitating. But all other disorders, and I think we can see it in ourselves too, if you're very, very stressed, you have difficulties remembering or learning things. And the same is in the case of pathology, where we have psychiatric disorders, there's always accompanied by deficit in cognition. For example, cognitive deficits in schizophrenia on the right-hand column, is, I think, the holy grail, which nobody has been able to tackle appropriately yet. And we are actually looking towards working in this area, hopefully also in the future. And what are the milestones in our project? You will see that in the clinical project, our lead candidate, GT002, which is just an acronym for our lead molecule, we've done through 3 types of clinical trials. Multiple ascending dose study, what came after a single ascending dose study, SAD study in humans, and we're currently doing the 3rd clinical trial in healthy volunteers, so-called target engagement study, where we look at EEG, fMRI, which is electroencephalogram and magnetic resonance imaging, and the effect of our candidate drug in healthy volunteers in order to see can we detect a pattern, and I'll come back to that later, that we can match it to the disorder for the next step later on this year, then going into patients and looking at the effect of GT002 in patients, which at the end of the day is the icing on the cake, which is the thing that you want to prove that your candidate actually has a beneficial effect in patients. This is what we want to get to. And for that, we've built about our, if you wish, the groundwork, the fundament for all this with these 3 clinical trials, and we've shown this drug is actually very, very promising, and most importantly, it is very, very safe and well tolerated. And I'm going to go on about this a little bit. I'm going to go in this particular presentation a little bit on why we've chosen EEG, electroencephalogram, as a biomarker, as an indicator of the activity that our drug actually does something in the brain that we wanted to do. And in this particular cartoon, it looks very impressive that you have a person with so many electrodes on the head. In practice, this is nothing more than a cap people are put on, and then you can do the measurements of the electrical activity in the brain. And to put it very crudely, the brain is actually an electrical machine. It communicates via electrical signals and chemical signals. You can then, of course, categorize these signals in different groups, alpha, beta, theta, and so on. And this is what you do with an EEG recording. EEG is non-invasive because all you do is you put on a cap. And it is, of course, very, very sensitive, and you can distinguish patterns in the EEG that become interesting for your effect that you want to see, and they're specific for the disease that you want to cure. So that's why it shows that we want to go with EEG as a biomarker for selecting our drug, and we want to see what patterns in the EEG and in the fMRI we actually induce with treatment with our lead candidate, and then matching it for patient group. That has been the clinical strategy for GT002. It will be the same one for all the other ones. This is our pilot project, of course. Our philosophy is that the EEG will reflect a deficit in the brain activity in different kind of disorders or different stress states. This can go anything from mild cognitive impairment to schizophrenia and other disorders, depression, for example. You will see changes in the EEG pattern. So what we do, we test the changes that we induce with our drug in healthy volunteers. Once we have mapped that pattern, then we can see how we can use that mapping into patients, which is then the next step. Here's an example what you can do. This is an activity map, if you wish. On the left-hand side, you have a screen and a normal and a disorder, and you can see that the EEG power, the EEG activity, the EEG energy, if you wish, in the different groups of EEG frequencies is different, and is differently affected in a mental disorder. So if you compare on the left-hand side, the patterns in the normal volunteer and the mental disorder are different. And that is exactly what we want to target, because now we're going to compare what does our drug do in the normal EEG pattern, and then we can match it to see how do we want to restore a normal EEG pattern from a diseased pattern. So this is what I have in the next slide. So you can see on the right-hand side now, you have again the same, a normal on the left-hand column, a mental disorder pattern, group of patterns in the middle, and what we want to do is GTX, which is generic for any of our drugs. What we want to, of course, do, we want to treat it again, and then we want to see that we can match the pattern on the very right-hand column and the left-hand column as much as we can. It will not be perfect. This is not a miracle drug, of course, but this will guide us in the way to go to see an effect. And it's a very, very straightforward philosophy, really, and it's a little bit a key and a lock, if you wish, as a principle that what you want to do, you want to match the pattern that you have in a disease state, and you want to restore it to a healthy state, giving you the drug. So it's basically very simple. If you have somebody that has high blood pressure, you would test our drug to see that it actually lowers the blood pressure in healthy volunteers. then you can go ahead and do the next step, and you have a good chance that it will lower the blood pressure in people that actually suffer from that. In the same way, you want to actually restore the brain activity, the brain patterns back to normal with our drug and measured by EEG, for example. So I think that is the key, and I think it's a very compelling, very straightforward way to match complementarity between our drug treatment and a restoration of a function as measured by EEG or imaging techniques, which I'm going to show you later. And this is going to be our way forward as a biomarker, as I mentioned earlier, for the EEG. So it's a little bit a technical digression, and I don't want to turn this into a scientific seminar, but I think that a number of people are actually interested in which is your way forward at Gabather as compared to others. We're approaching phase 2 with the lead candidate, as you can see, GT002 in the top row. For cognitive impairment, we are very much interested in, because of reasons I mentioned earlier, it's very ubiquitous symptomatology in different diseases. That means you can use it for a number of indications, and we're looking for partners, collaborators, license takers, now that we're at the borderline somewhere between phase 1B and phase 2. And it's a little bit semantics here, because phase 2 is supposed to be patients, but on the other hand, we are already now looking at the drug effect of our candidate drug and not only at safety. And of course, never forgot, but we are a reasonably small company. We do have a pipeline compound, and we are a bench to bedside company. So we have a lot of compounds on the bench who are waiting. Actually, they're waiting for me to go there and actually develop them and do it together with the team. Key insight from our studies that we have so far: we have a drug that is pharmaceutically developed. It's orally available. It might sound trivial. In pharmacodynamics, it's an absolutely key. It's well tolerated. It's very safe. We have not seen any harmful effects in any of our healthy volunteers so far. We can upscale the clinical material at a reasonably good cost efficiency, and we got the first form of our project developed, and actually it was a big breakthrough for us, and we actually patented our pharmacological form of the GT002. And it is a first in class. It is a new molecule in this arena. The GABAa receptor is a well-known drug target, and it's a very promising one, because there are many quite successful drugs in anxiety, in epilepsy, for example, that use the GABA receptor because it's just an important system. Our clinical phase 1 program is ongoing. Our phase 2 studies are planned for the end of the year. The pipeline is not forgotten. We've been very diligent with our patent and IP material and our IP work. So we have patented not only the composition of the compounds, but we've also patented the synthesis, we patented the formulation, and we patent the medical use. We're adding layer after layer of layer in the IP for our project. And the last thing I want to leave, I think this is one of the next to last slide. Currently ongoing, third clinical trial is a target engagement study. We're looking at EEG, as I mentioned previously, but we're also looking at fMRI, which is an imaging technique. The EEG will tell you what happens in the brain with a very quick time resolution. The fMRI will tell you where in the brain that happens, which is as important as the first information from the EEG to get an overall picture. And we believe that it is indeed a window into the brain, or a window into the brain activity, and that's exactly what we want to show and to prove also in the target engagement. Yes, our drug is safe. First hurdle taken. Next one is it goes into the brain. That hurdle we've also taken. So the third one is it modulates the brain activity in the way we want it to do. So it's promising for treating patients. At the end of the day, that's the key thing that we want to do. So our particular trial right now, this person is being pushed into the fMRI tube, basically, and measured the EEG at the same time, which is technically not unchallenging, to put it nicely. So it's a crossover study. So we're testing our compound. We're relative to placebo, of course, relative to already known compound. So we know both that the study is well done and our compound will have an interesting effect. That is different to something that you already have now. And the results will come by the summer. There's been a lot of both technical and logistical challenges for the last actually many months. but we are almost there. And I think it's very promising. And with the third clinical trial, we are very confident that we can go towards into patients, again, the final goal, and of course, looking for partners at the same time to run either clinical trials together with us or in combination with us, or to take over a license. We are very flexible in that respect. And I think I just want to, it's been very technical today. Last but not least, what is the market? What is the business in this? There's a very big market, as I mentioned earlier. And I always take one of the key examples of a deal, an agreement that was made, actually quite some time ago, and it was between Biogen and Sage Therapeutics in the US, worth 1.5 billion, I think. And the key thing is that this compound, Sage 217, is a small molecule also that targets the GABAa receptor and the modulators, and they used it for a depression trial, which has its own advantages and disadvantages. But the key story is that a lead candidate drug and a licensing agreement in this area can generate this kind of revenue, which I think is, again, to go back to the beginning, if we ever get anywhere close to that, I'd be more than happy. After all, I mean, I'm back in Lund, and the project started at the Chemical Center many, many years ago. It would be a great journey to finish it here too with this kind of agreement. And I think this is the last one. No, I still have one last thing on this particular last slide, is that, of course, since we're in a cognitive deficit and cognitive disorders area, what has been interesting is that there have been quite some requests regarding long COVID effects, because the effects of long COVID, now that the acute COVID wave is over, will become a next wave of people that are suffering from long-term COVID effects. And one of the long-term COVID effects, I'm not an MD, but one of the long-term COVID effects is, of course, cognitive deficits, i.e. people have problems with memory, with concentration, with learning. And that have been requested by us, because we do have clinical trial material that is safe by some networks. If we would want to work together with them in order to see if we can use GT002 with it for this kind of disorders too, which I think will be quite relevant for the next couple of years. So I think with this very last slide, I leave it there, and I just want to point out that we are a very small team. Actually, the whole of Gabather is on the left-hand side of the management group. So the reason why we've actually been able to get so far, if I may say so, is because this is a very good team. And if I look at the names of the three of us and calculated together how many man-years in drug discovery we have, we are approaching almost 100 years, which gives you a hint of how experienced we all are, actually. And I think with that, I have my very last word, which is thank you, and thank you for the presentation, and thank you to BioStock for being able to present here today.
Thank you so much for your presentation, Michael. You mentioned the fact that most mental health issues, at least the ones you presented to us here today, involve problems of cognition. Why such a big focus on cognition?
Well, cognition and cognitive deficits are directly related to, for example, the aging population that we have all over the world, not only Europe, also China, for example. Alzheimer's disease, key pathology is cognition deficits. But if you think about aging, even in healthy aging, we all know that the older we get, the more cognitive deficits you have. So it's huge. And another effect is, of course, that society today, with an information overload, there's a lot of stress in the background, and stress would definitely have an effect on cognitive abilities for everybody, and also including, in this case, not the old, but also the younger people.
Mm-hmm. And to follow up on that, what is the link between the GABAa receptor and cognition?
Well, GABAa receptor, since it's involved in almost all brain activities as the regulatory inhibitor, it's involved in all cognitive processes. It's present in all areas in the brain that also regulate cognition. So this is, of course, both a plus and a minus, that the GABA is so important. So you can put it as a cause or an effect of almost all pathology, and definitely also cognition.
Mm-hmm. In your presentation, you also mentioned the fact that there are other treatments for mood disorders being marketed that target the GABAa receptor. How does GT002 differentiate itself from these?
Yeah, well, you know, regarding mood disorders, the project on that is a completely different molecular structure, and it's a much easier pharmaceutical formulation, and it's another molecule structure altogether. You know, targeting the same area. So, if you want to compare it, I don't know, with an airport, you know, it's a completely different type of plane, but it's landing in the same place. So it is. The GABA receptor has been a drug target for many years, and the main area has been, of course, anxiolytic effects. Again, where we can see that anxiety is a component of almost all psychiatric disorders. So it's a key neurotransmitter. Again, I know I'm repeating myself, but you know, which is both for good and for bad, because you have to be very careful, and that's why we did so many clinical trials, just to establish safety and tolerability, because you're working with a key element of the brain homeostasis. So.
And finally, regarding the target engagement study that you mentioned in the presentation, you presented some preliminary results so far. What's the main takeaway from these results?
Well, the main takeaway is from the EEG data that we have. We will follow up with a very small group of volunteers with fMRI data. So we'll combine the EEG data with the imaging data, the functional data with the imaging data, and that then we'll finalize the study in the summer. And then we will know that we have a drug that's already available. It will go to the brain, and it modulates the brain activity in an interesting way in order to partner the project and to move towards the clinic. That's the key takeaway, and it's a big takeaway.
A lot to look forward to. Well, thank you so much, Michael, for joining us today, and we look forward to following Gabather in the future.
Thank you for having me, and looking forward to coming back, actually.